Welcome! Thank you for choosing ScienCell's GeneQuery qPCR Array Kit. This video will detail how to analyze qPCR array data by using ScienCell's Gene Query Analyzer. This tool was developed based on the comparative CT method, which uses endogenous reference gene and control sample to determine the relative quantity of target gene in the sample. If you have further questions, please contact ScienCell's technical support. Okay, let's begin. ScienCell's Gene Query Analyzer has 4 sections: Select Project, Input Data, Analysis Plots, and Output Table. We will explain each section in detail. The first section is to gather basic information about your project. Here, you should select the GeneQuery qPCR array kit you are using by either product catalog number or species. Alternatively, you can customize the 96-well plate with your own layout. Here we select GK139 GeneQuery Human Breast Cancer qPCR Array Kit as an example. By selecting this product, the genes layout of this qPCR array will be shown in the table below. Then, we want to indicate how many qPCR array plates are used in this project. Here we select 4 for example. Now we are going to explain the next section, which is Input Data. In this section, there are 3 information columns. The first column indicates the array position ID, which is row-oriented. The second column marks which genes will be used as endogenous reference genes. We might want to change the selections as needed. The third column lists the name of genes at respective positions. Now, we need to input the CT value for each plate and genes in the right positions. There are 3 ways to input the CT value data generated from ScienCell's GeneQuery qPCR Array Kit. First, we can simply type in the data one by one. Second, we can paste a whole plate data by clicking this button. To doing this, please refer to your qPCR Instruments data analysis software for data analysis and export. After you export the original data from qPCR Instrument by row-oriented, as an example, select all the CT values and copy to the clipboard. Go back to the Input Data section, click the button to instantly paste your data. Third, we already have the data from all plates in one spreadsheet as shown. We can copy all of the data and click the button Paste All or press the Paste hotkey. Okay, now let's scroll down and check whether the data is complete. The next step is to group the data. For example, we have duplicate results for control sample and duplicate results for the target sample. Hover on each plate and group them by selecting the desired data set or ungroup the plate from the current data set. The control set will be used as control sample in comparative CT method. Now, we can go to the next section to check the analysis plots. Here we provided a commonly used plot to show relative quantity of genes in the array. The data points dropped in this green area is considered as an upregulated gene compared to control sample. The data points dropped in this red area is considered as a downregulated gene compared to the control sample. Hovering over each data point will show the detail including name, position, and delta-delta relative quantity of the gene. By right-clicking on the plot, we can copy or save this image without interactive elements. The last section is the Output Table. We can inspect and copy process data in this table. The first line shows the selected endogenous reference genes and respective position ID. The second line shows the data we want to display in the table below, including the delta CT value, delta relative quantity, delta-delta CT value, and delta-delta relative quantity. The third line provides 2 formats of error, standard deviation or min-max value. After scrolling down and up to inspect our data, we can click the Copy All Data button and save all of the data showing here to the clipboard. Lastly, if desired, we can paste the data to a spreadsheet and begin further data analysis. Now we have finished all the processes using the ScienCell's Gene Query Analyzer. We hope that this online tool can facilitate the data analysis of ScienCell's GeneQuery qPCR Array Kit and saves your time from repetitive calculations. If you have further questions, please don't hesitate to contact ScienCell's support team. Thank you.